All right, so we got to this slide here where just kind of recapping what we said about different areas of the tubules and how different substances are reabsorbed or secreted, really in the broad scheme of thinking about how we're fine-tuning that filtrate. So here we're thinking about non-regulated reabsorption happening in the proximal tubule. So right after we leave the glomerular capsule and we enter this first part of the tubule, the proximal convoluted tubule here, this is where we see non-regulated reabsorption. So all of our ions, excepting hydrogen ions, are aggressively reabsorbed here. We're seeing glucose being reabsorbed, nutrients being reabsorbed, amino acids, and so on. And then a lot of water being reabsorbed here as well, but again, in an unregulated manner. If we think about the distal tubule and the collecting duct, we see regulated reabsorption, and we'll talk about the way that that is regulated going forward, but there are hormones that influence regulation in the distal part of the tubule and the collecting duct. And then we also see secretion of some substances as well. The loop of Henle is where we really see water conservation. So water is being reabsorbed here, specifically the descending limb. Solutes are being reabsorbed on the ascending limb, but this is how we conserve water. And we'll talk a lot about how the loop of Henle is really important for concentrating the urine and pulling most of that water back, but really making the urine as concentrated as it can be. And so in other species, we'll talk about this going forward, but desert rats, for example, camels, for example, they have really long loops of Henle, and that is to help them conserve even more water because evolutionary, they're not exposed to an environment where they have ready access to water as much as humans and some other species that have shorter loops of Henle. So the less access a species has to water, the longer their loops of Henle become, because the loop of Henle is really important in conserving water and concentrating the urine. All right. This is another really great study tool. So I talked about that initial schematic drawing that had the different segments of the tubular structures and even the glomerulus, and mainly introducing the functional aspects on that schematic. This is another table that's going to be a really good study tool that you can superimpose onto that drawing. So you can add some of the details here onto that drawing. But this just looks at what's reabsorbed versus secreted at each segment of the tubular structures. So for example, in the proximal tubule, we know that all of these things are reabsorbed, right? Glucose mainly, amino acids, et cetera, ions and so on, water. And then hydrogen ions are secreted. We'll talk about how this plays a role in our acid-base balance. In the loop of Henle, we'll add more details later, but generally speaking, water is reabsorbed across the descending limb. Solutes, sodium, chloride, potassium are reabsorbed across the ascending limb. And then the distal tubule, we see more reabsorption, but this is really regulated. In other words, we see hormones playing a role here. We see the nervous system playing a role here. And this is in an attempt to balance our plasma osmolarity. We also see potassium being secreted and hydrogen ions being secreted in both of these areas of the tubule as well. So another excellent table in terms of summarizing some of the main differences between different segments of the tubular structures. Okay, let's talk about the adaptation to the cells in these regions. And this is somewhat of a review. We introduced this idea the last time. Oops. Oh no. Did I press? Oh, okay. So we introduced this idea the last time, and we said that the proximal tubule is the mass reabsorber. There's 70% of sodium and water being reabsorbed here, 100% of glucose being reabsorbed, and we want to make sure that we understand that this is unregulated reabsorption. So as much as it's coming through, it's as much as it is being absorbed. We also see some adaptation of the cells here, the epithelial lining here. There's the brush border, and the brush border is mainly formed by the microvilli. So the apical parts of these cells have these foldings that are called microvilli. And these are different from cilia. I did have a student ask me previously about the difference between microvilli and cilia. So cilia are sticking up from the cells and are beating. Microvilli are not beating. They're just these foldings of the apical surface that really help to increase the surface area of the cells in order to really be an adaptation for absorption. And so the foldings here create this brush-like appearance in the very same way that we see bristles on a brush. The histologists that looked at these epithelial linings compared this to a brush. And so we call this area the brush border. There are also leaky tight junctions in between the cells. So there's some cell-to-cell openings here where we can have that paracellular transport. So some particles, substances can move right through the sides of these cells because the tight junctions here are quite leaky. And so this is another adaptation that indicates that we see unregulated or free reabsorption taking place across the proximal tubule. All right. And this is just kind of reiterated on this slide. I do want to point out, though, that we also see lots of mitochondria, and the shape of these cells are also another adaptation for reabsorption. So these are tall cells. These are called columnar cells. And these are the shape that we see when we have a lot of reabsorption of cells. We also see a lot of mitochondria, which provide the energy for all of that mass reabsorption. And contrasting that to what we see in the distal tubule and in the collecting duct. So the cells here do not have microvilli. They are shaped a little bit more differently. So they're kind of flatter. They're square shaped. These are called cuboidal cells, kind of square shaped cells. They don't have leaky tight junctions. In fact, the tight junctions here are tight junctions. So they're very secure, very tight fitting. There's no opportunity for paracellular transport, no opportunity for substances or molecules to move in between the sides of these cells. And so these are adaptations that are telling us that there's a very tight regulation of reabsorption here. So things are not moving freely the way they are in the proximal parts of the tubule, but there's very tight regulation here. We're also going to see that the cells here have specific channels on them to facilitate reabsorption. They also have specific receptor sites to bind some of the hormones that are going to be helping to regulate the reabsorption here. So the cells here look different, shape different. We don't have the microvilli, we don't have the leaky tight junctions. And so this is indicating to us that the reabsorption here is happening in a regulated manner, and it's not as freely as it is in the proximal tubule. Okay, we're going to just skip over that there. Getting to the loop of Henle, this is where we establish the gradient that is necessary for concentrating the urine. So I kind of touched on this earlier. The longer our loop of Henle, the more ability we have to concentrate the urine. That is to pull all of the water as much as possible from the urine and to lose as little water as is possible. Really hold on to that water for the purpose of, plasma conservation. And that's what happens in the loop of Henle. All right, getting into the final set of objectives here, we're going to talk about excretion. As a part of excretion, we're going to look at the clearance, which is the rate at which substances are excreted. We're going to talk about micturition, which is our final topic in this packet. And micturition is really much just voiding, how we control the functions of the bladder and of our sphincters in order to void the bladder and to eliminate urine from the body. Okay. Now, the excretion rate is essentially how much of our filtered substances we can add, how much of those substances is secreted, and then we subtract how much is reabsorbed. So whatever we excrete is really what's left after we have subtracted things that were reabsorbed and added things that were secreted. And so that is our rate of excretion. And so the amount or the total volume of substance that, or fluid rather, that is excreted, the total volume of fluid that is excreted is dependent upon the rate at which it's filtered, so filtered across the glomerulus, the rate at which it is secreted, so dumped from the blood into the tubular structures, and then the rate at which it is reabsorbed, so pulled from the tubular structures back into the blood or the plasma. Now, we can sort of deduce this relationship that if we see a solute appearing at high concentrations, higher concentration that it was filtered in the urine, we can come to the conclusion that that solute was reabsorbed, okay? Let's say I filter my glucose and I see less of that glucose in the urine. That tells me right away glucose was reabsorbed, all right? So if the amount that we see in the solute is less than what was filtered, reabsorption took place. The opposite is true. If the amount that we see in the solute exceeds what was filtered, then secretion took place. Again, if we think about, we're going to talk about PAH, or para-aminohepatic acid. That is something that is fully secreted. So it is showing up in the urine at a higher rate than it is being filtered. And so that tells us that it has to be coming from somewhere else. And that lets us know that it was secreted. All right. So looking more closely at this schematic here, we have filtration taking place across the glomerulus. Let's say we filter and we go from a solute concentration of 0.08 millimoles, and then we end up with a solute concentration of 12 millimoles. And then that gives us a water concentration of 150 milliliters, right? So that is what we start out with in the Bowman's capsule. So 12 millimoles of solute and 150 milliliters of water. As we move through the remaining parts of our tubular structures, we start to see these exchange processes take place. So we add 3 millimoles of solute by secreting certain substances, mainly potassium and hydrogen ions. And so that is going to increase the osmolarity of our solute in the tubular structure. So it goes from 12 millimoles where we started to now 15 millimoles because we have secreted 3 millimoles of solute. If we keep moving, we're going to see that reabsorption takes place. Now, reabsorption is not just solute, but also water. And so we reabsorb 145 milliliters of water, and we also reabsorb 6 millimoles of solute. So at the end of reabsorption, we also see a change in the osmolarity in the makeup of our filtrate once again. So now we go from 15 millimoles after secretion to 9 millimoles because we have lost 6 millimoles of solute. And then we go from 150 milliliters of water after secretion to 5 milliliters of water because we have reabsorbed most of that. We've reabsorbed 145 milliliters of water, okay? And so the remaining filtrate that we now excrete has an osmolarity of 9 millimoles of solute and a water concentration of 5 milliliters of water. And so this is how we can deduce whether or not filtration or secretion took place by looking at the amount of solute that's left in our excretion, in our urine at the end here, versus the amount of water that's left in our urine at the end as well. And on this table, we're just talking about clearance. And clearance, there's not a clear definition in your packet, but clearance is the rate at which substances are excreted. Okay, it's the rate at which substances are excreted after being filtered across the glomerulus. And so we can use the clearance rate of some of our ions and molecules that are listed to the left here to tell us about different exchange processes or to tell us about our GFR. So if we think about PAH, this is what I was speaking about earlier, which is para-aminohepatic acid, or PAH. This is something that is fully secreted. It's not reabsorbed at all. And so it has a clearance rate of 650 milliliters per minute. And so we can use PAH to tell us about renal plasma flow. In other words, it will tell us about how much blood is flowing through the kidneys because it is not reabsorbed, it is filtered and then fully secreted. And so all of it should end up in our urine. And so because it's moving from the vascular structures, because it's moving from the peritubular capillaries, from the vasa recta, and it's moving into our tubule, it's going to tell us a lot about the flow rate of blood that's moving through our kidneys, okay? And so clinicians will measure the amount of PAH in your urine, and that will tell about your renal plasma flow, okay? Now, PAH is an exogenous substance that we insert into the vasculature. It's not made in the body, so you're going to have to insert that into your blood and then measure how it comes out in the urine. We can also look at things like creatinine and inulin. Inulin is also exogenously introduced. It's a byproduct in plants, I think. It's one of those polysaccharides. And inulin has a clearance weight that is 125. So it's a really good predictor or a measurer or an indicator of what GFR looks like. It's neither reabsorbed nor is it secreted. And so it's going to give us a really clear picture about the GFR, about our glomerular filtration rate. And so renal clinicians and renal physiologists will introduce inulin, again, into our plasma exogenously, and then they'll measure how it comes out in the urine. And because it's neither reabsorbed or secreted, it's going to give us a really good indicator of what our GFR looks like, right? It has a clearance rate of 125, which is also the rate of our GFR. Creatinine is another thing that we look at in terms of somewhat predicting GFR. It's slightly higher than GFR because it is slightly secreted, right? So a little bit more of it comes out in the urine than our GFR, but it's also a good indicator to tell us if GFR is below the normal levels because we can think about how this is shifting in terms of that 140. So creatinine and inulin are two substances that can tell us about GFR, whereas PAH is a substance that can tell us about the renal plasma flow rate, how blood is moving through our kidneys. And so what this table is really helping us to see is that if we compare the clearance rate of certain molecules and certain substances to our GFR, that will tell us whether or not it's being secreted or if it's being absorbed or neither. If it's being neither secreted or reabsorbed, as is the case with inulin. If we think about our ions at the bottom here, they're being fully reabsorbed. And so we see very small amounts coming out, or very small weights in terms of the clearance rates of these particles. So essentially, if the clearance rate is greater than GFR, we know that secretion is taking place because it has to be coming from somewhere at a faster rate. If the clearance rate is less than GFR, we know that net absorption is taking place because it has to be going somewhere else, okay? So that's kind of what we can deduce from this table. All right, let's take a brief pause here and see what questions we have so far. Okay. All right, our final objective in this packet, the very end here, is to talk about micturition. And we said that micturition is the process of voiding, so emptying the bladder. And so we're going to talk about the different components of the nervous system that are involved in bringing about micturition. So micturition is the process of urination. It's where we empty or void the bladder, urine that's basically formed in the renal tubules. So after we fine tune our filtrate and we send it down by the ureters to the bladder, we basically store it in the bladder until it's time to be excreted from the body. Okay, so I am getting a question here about going over GFR and clearance relation. Okay, excellent. So just to touch on that one more time here, and that's a really good point because that's kind of the main thing that I want you to take away from clearance and from this table, is if we see a clearance rate of a substance or a molecule or particle that we can, like we can see here, our ions and some of these other substances, if we see a clearance rate that is higher than our GFR, that tells us that net secretion took place, okay? If we see a clearance rate that is the exact same as our GFR, neither secretion nor reabsorption took place. So that substance is appearing in the urine at the very same rate at which it is being filtered across the glomerulus, okay? If it's appearing in the urine at a rate that's faster than GFR, we know that there's another source of it entering the tubular structures. So we know that secretion has taken place, okay? On the other end of that, if we see a clearance rate that's less than our GFR, it's appearing in the urine at a slower rate than what we see it being filtered across the glomerulus, we know that it's going somewhere else, right? It's going to be reabsorbed. And so we know that net reabsorption has taken place, all right? So this is why inulin is such a great predictor of GFR, because it's neither reabsorbed or secreted. It's coming out at the exact same rate at which it is being filtered across the glomerulus. And so it has a clearance rate of 125, which is also the GFR, 125 milliliters